Hello. Okay. So yeah, I'm Charlotte, and I'm from Tobias von der Haar's lab in the University of Kent, United Kingdom. And I'm going to be talking to you about the work that I did throughout my PhD on how we can use these biofilms to enhance plastic degradation and how they're biotechnologically relevant. There we go. So Dr. Misa gave a fantastic introduction earlier with brilliant storylines, and it was wonderful. Mine's a bit more boring. I've just got a lot of stats for you. So in the UK, only 55.5% of our plastic is actually being recycled. And considering we're making over 400 million tons globally of these different types of plastic materials, that's not good enough. And only 10% of all plastic materials ever made globally have actually been subjected to some form of recycling, which is resulting in a lot of industries using these plastics and then them being discarded into the environment. So I'm going to be talking to you specifically about PET, but obviously there are lots of different types of plastic, and we're hoping that my system is applicable to all the different types of plastic. So yeah, once a piece of plastic has been used in this industry, it often finds its way into the environment through being discarded in landfill or just entering through overusage and the plastic breaking down. So once the plastic has entered the environment, what happens is it will fragment, crack, or the surface will become disrupted, and then these plastics become smaller and smaller and smaller, and that's where we get our microplastics, which then can further fragment into nanoplastics, and then we get our picoplastics. And one of the scary facts that have recently come out, so I started my PhD in 2020, and back then we weren't really talking about the impact it had on humans because we didn't worry about that. We didn't know that was a thing. But I can guarantee if you Google microplastics today, you'll see lots of studies saying that they're accumulating in humans, which that's only 4 or 5 years ago. And that's terrifying that we're now starting to see that they're getting inside of our bodies and we don't know what's going to happen to us. So this is because our recycling is not doing what it should be doing. So a lot of the time we're talking about this thing called a circular economy, and that's where a piece of plastic is made and it stays inside of the circle. And if that was happening, that would be great and we wouldn't have any of these issues. But unfortunately, the plastic is exiting the circle through usage or discarding. So what we're trying to do is figure out a way of how we can make our plastics circular. So, like I mentioned, there are lots of different types of plastic out there, and I'm going to be specifically talking about polyethylene terephthalate, or PET. It's actually fantastic to see that we've all got glass bottles in here, not plastic ones, otherwise I'd probably have a heart attack. But PET is made up of a terephthalic acid group and then an ethylene group, and there are over 500 billion PET plastic bottles produced each year, and only 19% of those are actually recycled globally, which is honestly not good enough. And most of these are the type of plastic that we will come into contact with. And additionally, polyethylene terephthalate is also found in clothing, so all of our clothes have some type of polyester in it. So it is not the biggest produced plastic globally, but it is one of the ones that humans will come into contact the most with. Oh, and also, in addition to that, it's one of the most researched for naturally occurring enzymes that are able to break down plastic bottles. And a lot of these have been isolated from plants because the enzymes that have been discovered are very similar to plant structures, and they're able to break down plants as well as plastic, which is interesting. So the enzyme that I'm going to talk to you guys about today is quite a famous one. It's called PET hydrolase, or PETase, and it was discovered in Japan in 2016, and it was shown to be able to bind to plastic monomers, polymers, and break it down into these different monomers, and then eventually these monomers here will break down into these even smaller monomers, which is terephthalic acid and ethylene glycol, and these are practically non-hazardous when released into the environment in small amounts. So we were really interested in how we could take this enzyme and take our own way of using it. So a lot of the time when people are producing this enzyme, they will produce it in bacteria and then do your standard protein purification and then have a really controlled environment for that reaction to happen in. So we're going into specific buffers with a specific pH. And this is time consuming and quite expensive and also not necessarily scalable. So we were interested in making it cheaper and easier to use. And by doing that, we were looking at producing it in Saccharomyces cerevisiae, but secreting it into the extracellular environments. That was the initial idea for my project was, okay, let's take Saccharomyces cerevisiae, because my supervisor is an expert in this field, and see if we can secrete these different plastic degrading enzymes. And what we were then interested in a little bit further down the line, so a couple of months into my PhD, was, oh, actually, Saccharomyces cerevisiae is actually all right at adhering to different surfaces. So if we can get them to form what we call biofilms on the plastic surface, will this increase our plastic degradation that's happening overall? And there are lots of different strains of Saccharomyces cerevisiae, as I'm sure a lot of you already know. And these strains can vary quite significantly in their phenotype. So we had a look at the different strains available to us, with our hypothesis of that engineered yeast biofilms should directly deliver the PET hydrolases to different PET surfaces. So the fact that they're adhering to these surfaces, they should be able to secrete the protein to the surface, and they should be a lot more concentrated than if it was just floating around in the media in those controlled reactions. So yeah, we started looking at the different strains that we had specifically available to us at the start of this project, which wasn't very many different types of strains, because previously we were just working in the standard lab strain BY4741 or S28AC. And we started comparing their ability to adhere to different plastic surfaces. So this experiment is actually the same as what Chloe explained earlier with her biomass assay. It's called a crystal violet assay, and it's essentially where you add this stain to your yeast or your culture that you've grown, and then incubate it with the stain, and then eventually elute it off with acetic acid, and then it will give you an absorbance value. And the higher the absorbance value, the more cells that had adhered to that plastic prior to washing. So we saw that we had this one strain here, which is used throughout the wine industry, that was quite good at forming biofilms. Yes, it was very variable, but it was a lot better than our standard lab strain, BY4741. So we then started to look at, could it form biofilms on PET materials? And we saw that after 96 hours, we were getting quite good biofilm formation here along the edges of our plastic. And what we defined those as was rougher regions. So essentially this was a piece of a plastic bottle that I got a hole punch and cut a hole in, so they were all the same size. And obviously when you're cutting something, you're leaving quite a rough edge, and that's what we were getting here. And we were seeing that the yeast preferred to form their biofilms in these rougher regions. So we'd identified the strains that we wanted to work with. So just to clarify for later on, is that we decided to work with BY4741 as our non-biofilm forming strain, and then Sigma 1278B as our biofilm forming strain. We had to design how we were going to then produce our protein in our yeast. So luckily, and fortunately later Tobias will be explaining this to you a bit better, is that we decided to optimize our PET hydrolase genes using an online platform that he's developed. So we codon optimized it to Saccharomyces cerevisiae usage, because I didn't mention this earlier, but the PET hydrolase was from bacterial origin, so secretion might have been less than optimal if we had just produced it as was. So we codon optimized it to our codon usage, and then we inserted it into our plasmid, which had a signal peptide that we'd selected with a HA tag for Western blotting. This is important because later that kind of slowed down my process a bit by using HA rather than another type of tag. And then we had it under the TDH3 promoter. And these plasmids are all available online on AdGene if you're interested in looking at them in more detail. So we were able to transform all of our strains of Saccharomyces, and we were able to produce the protein, which is 32.5 kilodaltons, quite nicely inside of our cell. We then looked outside of the cell in the growth media to see if we were getting any secretion, and we saw that we were getting relatively high levels of protein being produced outside of the cell, which is fantastic. However, what we noticed was that our molecular weight was a lot higher than we'd expected. So we were expecting a band roughly down here, well, actually down here. And unfortunately it was a lot higher. So this led us to believe that something was happening to our protein during the secretion process, possibly some type of post-translational modification. And we ran a screen using this online platform called MuSite Deep, which is where you can plug in your protein sequence and then look for every type of post-translational modification that could happen, and it will predict how likely it is to happen from a scale of 0 to 1. And what we saw consistently was that the highest confidence level was in N-linked glycosylation. So when we changed it from a 0.5 confidence value up to 0.9, so very highly likely to be happening, it was N-linked glycosylation. And then to confirm it was N-linked glycosylation happening to our protein, we treated it with an analytical enzyme called EndoHF. And this essentially just closes off all the mannose sugars at the asparagine sites, confirming that actually, yes, our protein was being glycosylated. And that's sort of where we left it with the glycosylation was, okay, we know it's being glycosylated. that's what's happening. That's what it is. So we decided to see, okay, we've got this protein, we're secreting it, will our idea actually work? So just to talk about how I actually set up these degradation assays, I take my pate secreting yeast and then put a plastic bottle in its culture, and then I leave it for however long I want to leave it for. And I incubate them at 30 degrees. Often my assays were about 96 hours. Sometimes I would leave them for a week. Sometimes I would leave them for less time. It would depend on like the deadline I was working to. There was no real logic behind that part. But obviously all of the results I show you, they are standard. I have standardized them, what I'm showing here. We obviously buffered the media as well to make sure it was pH 8, because Petase's optimal activity was between pH 7 and pH 9, and we showed that our Saccharomyces was best at growing at pH 8. And then to assess whether we had degradation, we had multiple different routes and different ways to analyze it. We used atomic force microscopy, which I will explain a bit more in a second, scanning electron microscopy, and then again, atomic force microscopy for our quantitative evaluation, and HPLC. So what is atomic force microscopy? I don't know if many people have heard of this technique. It's quite niche. So essentially, it's a biophysical way of analyzing things, and you have a laser that will go down onto the top of this tip, and this tip is going across your surface, tapping, essentially. And as it's tapping, it bounces off that laser and then sends signal back to a computer, which will give you values for different areas. So the lighter areas, or the neutral areas, are smooth through to light being raised on the surface, and then our darker areas are essentially holes and pits in the surface. So we checked our non-biofilm forming strain of Saccharomyces compared to our control, which had no protein being produced, and our Petases, and we saw that our surfaces are quite clearly different, and that we're getting a lot more darker areas happening on our surface. And then this was also consistent in our biofilm forming strain. We saw that the surface at the start stayed relatively smooth, and then we were getting more and more darker areas on the biofilm forming strain. And then when we compared the two together, we saw that overall degradation was a lot higher in our biofilm forming strain. We then confirmed this data with SEM and saw almost parallel the exact same results. So these are two different experiments done in triplicate, and we were able to reproduce it and analyze it using SEM. And then also why we decided to move forward and just use AFM over SEM in the future was because it's mechanically touching the surface. It means it can then give you actually quantitative values such as roughness. So using the roughness values it gave us, we were able to quantify how much degradation was happening overall. And we saw that in our biofilm forming strain, it was a lot higher and a lot less variability in overall surface roughness in our plastic coupon that we'd incubated with ourselves, which was fantastic. So at this point, we had confirmed that biofilm formation indeed does actually increase degradation, but we wanted to see how far we could push this. So we started looking at how we can encourage biofilm formation. So I come from a research group that looks at Candida albicans, and they're really good at forming biofilms. So when they see my cells, Saccharomyces, they often use it as a negative control for biofilm formation, which makes my work look really rubbish when I'm with them. But we wanted to see if we could push and drive forwards and make Saccharomyces actually a suitable candidate for biofilm formation. And we decided to do this by incubating our yeast in different concentrations of glucose. So lots of studies out there had said if you increase glucose, you get higher levels of biofilm formation happening on a surface. I wanted to see if you decrease the glucose available to them, could that also encourage biofilm formation? So standardly, Saccharomyces grows in a 2% concentration of glucose, and we saw that once we decreased the amount of glucose available, we were getting higher levels of biofilm happening in our non-biofilm forming strains. So that's really important to notice, is moving beyond this, I stopped working with that biofilm forming strain because I wanted to see if I could push a non-biofilm forming strain of Saccharomyces. So yeah, we were able to see higher levels of biofilm formation happening in our 0.5% glucose, and you can see that also with the crystal violet staining. I wanted to make sure that that wasn't then killing off all my cells, and we saw that actually, yeah, there was a lot more death happening in the 0.5% glucose concentration, but there's still 50% alive, so we'll take that as a positive, as 50% of the cells are still there. Moving beyond this, we wanted to see whether this change in glucose available would actually impact the amount of protein being secreted outside of the cell. And interestingly, it had no impact on the cell and the production of the protein. And also in another plot that I haven't actually shown here, we were getting a reduced amount of glycosylation happening to our protein because there's less glucose available for that process to happen. So that was really interesting. And then in addition to that, something that we came across by accident was that the buffering capacity of our yeast increased with the amount of decreased glucose. So at 0.5% glucose, we were having our pH stay at a much higher level. And if you remember earlier, I talked about how I was buffering my reactions pH 8. Interestingly, after about 24 hours of our experiment running, the yeast had acidified the media so much that it would drop down to pH 2, between 2 and 4, which then essentially renders our protein useless after those 24 hours. So despite how long I kept those experiments going, they'd stopped after 24 hours. We then looked at that in our 0.5% glucose concentration, and it only dropped after 24 hours to pH 6. And then I did that for a week, and it just stayed at pH 6. So our pH is staying much higher throughout, which is awesome. We then, of course, looked at whether that had any impact on our activity, and excitingly, it did. We had a lot more degradation happening that we confirmed through AFM once more in our 0.5% glucose compared to our 2%, which was our previous reactions. So overall, you can see that the surface is a lot darker. You will notice that there are more white regions, so different to what was shown earlier. The white regions are areas of raised on the surface, so that's just salt deposits. So we're getting a lot more gunk in the media, which isn't great. But when we're doing these roughness calculations, we will randomly choose areas on the surface, but I will also try to avoid putting a square right in front of where the salt crystals are, because then it will just give you a really high roughness value and won't be accurate to what we're looking at. So yeah, it was confirmed that we had almost double the amount of degradation happening, which was fantastic. Beyond just this one enzyme, so I've just talked to you about one of the PT hydrolases I've worked with, which is just PETase. I've moved on to express 7 different PT hydrolases in Saccharomyces through secreting it into the extracellular environment. And we saw that with these different, they all had the same signal peptide, all in the same plasmid, all under the same promoter. We were getting different levels of protein being produced in the extracellular environment. And we have PETase, thermostable variant of PETase, and then hot PETase. These have all had mutations done, not by our lab, by other labs, that have changed the actual core protein sequence, which I believe is reducing the amount of glycosylation sites available to happen, which is why we're getting more protein being secreted. And then we have these other enzymes here called LCC, ICCG, and we're getting, obviously, they're 2 of the same, and then this one's more thermostably variant of it, and we're getting more protein produced. And then we also looked at a couple of other ones. So this one's from Antarctica, this one's from clothing, and then I don't really know where this one came from, but it was another good candidate back in the day when we chose it. And we saw that there was lots of differences shown in the amount of degradation happening. So we have our control here, and then we have all of our different surfaces, and we can see in PHL7 in this particular assay, we're actually starting to get holes in our surface, and the same here for BHR PETase, which was the first time that we were starting to see these proper holes rather than just pitting across the surface. And then I transformed, so if you remember the graph at the very start, I had 3 different strains. I transformed all 3 of them, but I've only shown data for 2 of them to sort of just get a point across. The data doesn't necessarily matter here, but between the different strains of Saccharomyces, we were getting different levels of activity happening. And interestingly, all of these were done in 0.5% glucose, so encouraging that biofilm formation. Our best candidate was the original PETase that we started working with at the start. So the thermostable variants actually had no impact on degradation overall. But yeah, our initial idea with this was to sort of have multi-enzymes, so have our Saccharomyces producing different enzymes. So to do that, we would have to, I'll come back to that in a second. We would have to integrate the protein into the cell, which I will talk about in the future. So one final thing that I always get asked at my talks is how much protein are you secreting? Not very much, unfortunately, which was really devastating for us when we had to quantify the amount of protein we were getting. So we're like, okay, this is good. We've got good levels of activity. This is working really well. and it's all going fantastic. But unfortunately, after doing some densitometry, which might not be entirely accurate, we were seeing that only 400 micrograms of one of the proteins per liter was being produced. And then in one of the other PT hydrolases, we were only getting 2 micrograms. I will say, I have to see the positive side of it is that we're getting such small amounts of protein, but we are still getting quite good levels of degradation happening overall. So actually our biofilm is really impacting the amount of degradations that's happening. Because if you were to put this amount of protein into one of these perfect reactions, you would probably see no activity at all. So yeah, as I mentioned, we wanted to have our multi-enzyme biofilms, but to be able to do this, we had to start making strains of Saccharomyces. So all of the expression I've talked to you previously about was extrachromosomal. We then started looking at making our strains that were producing these different enzymes to then deliver them all to the surface and hopefully just completely eliminate a piece of plastic, which is an ambitious dream that we had at the start of my PhD. I've lost the thing. Oh no, I haven't. Here it is. We used this paper here by Laffery from 2015 that has a fantastic protocol to CRISPR-Cas9 and Saccharomyces cerevisiae, which is just essentially a one plasmid reaction. So this plasmid will have your CRISPR-Cas9 in it, and then it will also have your guide RNA inside of the plasmid as well. So then in the Saccharomyces transformation, all you need to do is put this plasmid into the reaction, plus your gene of interest with homologous wings on each side, and we used 500 base pair upstream and then 500 base pairs downstream, and then you just transform that all into your Saccharomyces, and you should have CRISPR happen within about a week's time. And we did this with one of our enzymes and saw that compared to our extrachromosomal, which was the original protein we were secreting here, compared to our integrated protein, we were getting a lot more protein being produced once we integrate it into the Saccharomyces genome. There is one core thing that is different, is that in our extrachromosomal plasmid work, we were using a different signal peptide to what we integrated at, as that wasn't native to Saccharomyces, the signal peptide. So with higher levels of protein being produced, we wanted to then check if we were getting higher levels of degradation happening. And this was the first time I was like, oh, we're actually going to get rid of a whole plastic bottle. It didn't go that far, but it was the first sign that we were like, okay, this is going to work really, really well. Because we were getting almost black areas of surface, which was fantastic compared to our other areas on the other non-integrated strains, still looking relatively smooth and comparative to our controls. And then once again, this was confirmed by our roughness measurements, where we saw that we had a much higher value, and this is higher compared to every single assay we did throughout my entire PhD. So we showed that actually integrating the protein into the Saccharomyces genome at the specific site that we chose could be a really good step forward in actually completely eliminating plastic waste. So furthermore, the application value of it all was to see if we could then have these big bioreactors where we would dump our plastic in and not have to worry too much, and then out would come with the degradation products I talked about at the start. And we started playing around with Saccharomyces in our fermenters, and unfortunately we had issues with our flocculation. So the talk before talked all about how flocculation is positive, but actually in our specific experiment it was really bad, and it kept clogging up our fermenters and we weren't able to do what we wanted to do. And annoyingly, the strain that I'd integrated PETAs into was a really highly flocculating strain of Saccharomyces. So that just resulted in us having to make our own fermenter. And essentially it was a really simple system where I had a glass bottle with our plastic suspended inside of it like this, and then we had a media reservoir, and it would just pump fresh media in over time. So essentially the same as a fermenter, just a bit more, less of a risk that I could break it for other people. And then I guess to summarize, the overall thing that I've shown from this work is that Saccharomyces can secrete PET hydrolases into the extracellular environment. We can now confirm that biofilm formation can increase surface roughness, suggesting more efficient degradation overall. And then if we adjust the abiotic environment, we can increase biofilm formation, which then increases surface roughness, protein secretion, pH stability, and reduce glycosylation. And then also PET hydrolase degradation will vary between enzyme and strain of yeast that it's expressed in. And then if we integrate these PET hydrolases into the genome, and we can see higher levels of degradation overall, which is fantastic. And what's really interesting now is I've just started my postdoc in Tobias's lab still, but we started having companies and applying our technology to their work, and we've started recently working on seaweed lines to see whether we can deliver enzymes to these seaweed lines to break them down, which has been a really exciting shift and application of my work. So yeah, a big thank you to absolutely everyone that's been involved. It's been a very multidisciplinary team that's helped me achieve all of this, and in particular Tobias Wonderhaar, who will be giving a talk shortly, and my co-supervisor at Portsmouth University, Andy Pickford, and SocoBio for funding my work for the last 4 years. Thank you.
Thank you, Ms. Charab. Okay, so we have 4 minutes, maybe 2 questions.
Thank you for sharing informative talk. I just wonder, like, do you have any explanation why different strain of yeast can make different enzyme with different activities?
Yeah, I think the strain side of things will be linked back to the biofilm phenotype broadly, is that they they might not be adhering to the surface as well. I think they were all producing it at relatively similar levels. So then it has to just be something that's morphological. And I reckon it's probably due to the like surface contact of the yeast and the plastic, is what I would guess.
Yeah, that makes sense to me.
Thanks.
So you try to improve the biofilm formation. So do you ever try to like the, how to say, like the adding some element like manganese, copper, or things like to increase the biofilm formation?
Yeah, so in literally the first year of my PhD, I was trying to produce one of the flow proteins. So I was trying to use flow 11, which is like cell surface adhesion, and I just had no luck of transforming it into my yeast. I think if I could do that genetically, it would probably work really well and help a lot. I just wasn't able to make it happen.
Yeah, previous I used Bacillus subtilis, so
Nice.
that is the research study that they use the vary the concentration of the manganese, and they found like a certain concentration is can like enhance the formation
sure.
of the biofilm.
Yeah, iron like helps with bacillus, right? Like
Yeah.
if you increase iron. Yeah. So I tried to pitch this project to my supervisor and say, can I stop working with Saccharomyces? Please, can I work with some other type of organism? And bacillus was one of them because obviously they're really good at forming biofilms. So that was something that we started to look at, but we haven't gone any further with this. We're working with other bacteria now, and we did look at other like strains or species of yeast to try and do this as well. Yeah.
Thank you.
That's okay.